All right, boys. Assalamu alaikum wa rahmatullahi wa barakatuhu. Good morning, everyone. I hope everybody is doing well, inshallah. And today we will be doing the topic of enzyme. I'm on the canvas right now. The learning objectives are with us. We have recently studied about the biological molecules that are carbohydrates, fats, and proteins. And now is how they're going to be digested, and what are enzymes and what is the role of enzymes, how were they named, how do they work, and what are the different properties of them. So we're going to be studying about these. So this contains a worksheet over here with the links. So we're going to go through a small activity. We're going to go through the flashcards. Anybody who would like to define what is an enzyme? If anybody remembers from the previous lessons, we have seen a few videos on the topic. Anybody who could remind me that what was an enzyme? You can answer through chat or you can raise a hand. I can give you the mic. Yes, Mr. Anderson. What do you think? What is an enzyme? And I'll give the mic to Awais. Yes, Awais.
Mr. Enzyme is a substance. Um, it speeds up chemical reaction.
Excellent answer. Absolutely right. It is a substance. It's actually a protein that can speed up a chemical reaction. We call them a biological catalyst. A catalyst is the same thing that is speeding up a chemical reaction. So excellent answer, Awais. Very good job. Thank you. So we'll go to the next card. Catalyst. We just said what is a catalyst. Ghazi, no, this is too simple. I'll give the mic to Anderson. Anderson, you don't even have a mic. Why? Hatim Al-Amri, would you like to? Oh, he doesn't have a mic too. Very good. Mansoor, would you like to tell
Hello,
us
Mister.
how are you?
Yes, Mister.
Would you like to tell us what is a catalyst?
So a catalyst is a
And you muted yourself? Yes.
no, Mister. Something is wrong with the network.
Sorry, no problem. Let's hear
I
your
can't.
answer.
Okay, so what do they call it? So a cat. Mister, can I
You can chat, you can answer a text, no problem.
Okay, Mister, can I close the network? Open it again.
Yes, sure, do that.
Okay.
Okay, so we'll go to Awas. Awas, you just answered it. So I'll give a chance to Abdul Aziz. Okay, so nobody's gonna go through this, so I'll just open it up. The answer, it speeds up the chemical reaction. I know, Ghazi, you know this, so let's go to the difficult activation energy. Now this is something. Yes, what is activation energy, Ghazi?
Mr. Wallahi, I forget, but I know it. That's like familiar word.
Try to say anything that comes to your mind.
Mr. That like energy is made from Allah, like a glucose. It comes from the sun.
Yes, that is true. So what is activation energy? Something which is activating.
Mr. I know it's something like a give energy, active energy.
Remember, when we have to start a chemical reaction, we have to give some energy to let it start.
Yes, sir.
That is activation energy. The energy needed to activate a chemical reaction.
Okay.
All right, good. So Awaz has a very good question. He says that, what is the difference between enzyme and a catalyst? Who would like to answer that? What is the difference between a catalyst and an enzyme? Yes, Ghazi.
Mr. Enzyme is a biological catalyst.
Yes, you are right. So let me explain in simple words for Awais that it is in the living body. If a catalyst is in a living body, we call it an enzyme. And if it is outside the living body, we call it a catalyst. As simple as that. Wa alaikum assalam wa rahmatullah Tahir. How are you? So let's go to substrate or substrata. Some people like to say it. Who would like to answer this? Via, are you with us? Via, I'm going to give you the mic. What is a substrate? Substrate. No, he doesn't want to answer. Basim, are you with us? No Basim present.
Yes, mister. I just came. I was in the bathroom.
Okay, welcome back.
You're welcome.
So, what is a substrate? Do you remember anything that comes to your mind? I know Ghazi, you know it. Basim also knows it, but he has to recall it. Where did Basim go? He put himself mute. Basim, do you want to answer this question?
Yes, mister. What was the question?
What is a substrate?
A substrate. The molecule that the enzyme break it.
Exactly. Perfect answer. Well done. Full marks for Basim. Good job. It is the molecule on which the enzyme is going to act. It's called a substrate. So let's check the definition. A reactant of an enzyme catalyzed reaction. Yes. So enzyme substrate complex, we didn't do this, so let's leave it. Product. Yes. What is a product? Abdurrahman Tudsi or Mansoor. So we'll start with Mansoor. Yes, Mansoor, how is your network now?
Hello, Mister.
Hello.
Yeah, my network is now. Inshallah, is good.
Inshallah. So what is a product? What do you think?
I think this product means a species formed from a chemical reaction.
Yes, it's not going to be a species, it's going to be a substance or a specimen, but your answer is correct. That is something as a result from the enzyme and substrate working together when they produce something that is a product. So good job. I'll accept that answer as well. So let's see the chemical substance formed as a result of a chemical reaction. It could be any chemical reaction, like outside the body or inside the body. So let's go to the next. Competitive inhibitor. No, we didn't study that. Inhibitors is not active site. Abdul Rahman Junsi. So after that, Abdul Aziz, I'll come to you. Abdul Rahman, did you get the mic? Abdul Rahman is, yes. Hello?
Hello?
Your voice is very low. Can you put the mic in front of you?
Hello?
Yes, feeble voice, but all right. Yes, Abdul Rahman. Yes, what is an active site?
Hello?
Yes.
An active site is a region where enzymes and molecules bind.
Excellent. Absolutely true. So good answer by Abdul Rahman. He said that the active site is the location, is a place where the substrate and the enzymes combine. It's actually on the enzyme. It's the place where the substrate will come and get attached to it. So excellent answer. Good. So let's go to Mansoor. Mansoor has a question, and Ghazi, I'll come to you next. Yes, Mansoor.
Mister, can you show us all the questions and the answers because I want to write it?
Exactly. What's your good name, Mansoor? This is the site. It's on the canvas. Vocabulary flash.
Yeah.
So once you open it, you're going to have all the questions to
Yeah.
answer. There are 17 questions. We are on question number 9 right now. So no, you don't have to write it. Just save these questions on the Word file.
Oh, so it's in Canvas.
It's in the Canvas. And from the website, you can copy paste these questions and paste it in a Word file and revise it when you're ready for the exam. Right?
Okay.
All right. Good job.
Yes.
So this is the part of an enzyme of no, it's not antibody. It's a part of the enzyme where the chemical reaction occurs and where the substrate gets connected. Yusuf Jimjoom is in the waiting room. Oh, sorry. I have a lot of boys in the waiting room, and I was looking at the waiting room and I thought there was nobody here. Okay, so would I? Okay, good. Alhamdulillah. So we are recording this session. Alhamdulillah, again, I had panicked. So Yusuf, sorry about Yusuf. And sorry about the guys who were in the waiting room. Yes, Yusuf.
Mr. What did I miss? Because I was in the waiting room waiting.
Yes, I'm sorry for that. We were going through the revision session. We have flashed about 8 cards. This is the 9th one. We are discussing about the enzymes and what are enzymes' job, what do they do, and these one. These two competitor inhibitors we didn't do, so we skipped it. We discussed what is a product, and we skipped this one because this is not what we have done. So we have done like substrate, activation energy, catalyst. So about five of these flashcards we have discussed.
Mr. But I have a question. When are
Yes.
we gonna start revision now?
No, the revision is gonna be from the 8th week. 8th week, yes.
Like in 2 weeks, right?
Yes, we are on week 6, so 7
Yeah.
will be further, and the 8th one we'll start revising, inshallah. Hopefully the 9th week is the exam.
طب I have a when we start revision, I want to ask because when we took about combined sciences, and I got half of the things about monosaccharides and disaccharides, but I need more. Yeah.
Okay, inshallah, we will discuss that. Don't worry. So this is right now more like a revision session as well because we have done enzymes, and let's try it again. Allosteric site, we didn't discuss this, so we'll skip it. Factors that affect enzyme function. This is important. Who would like to mention this? We have a chat. Yusuf, if you know, you can share. Inshallah, by the next week, I'll give you the revision pages. Okay. So Ghazi, let's hear Ghazi. What are the factors? What are those things that can...
Wait a minute, Mister. Just wait a minute. My cat will die.
Who will die? It's a matter of life and death. So, okay. So let's go to anybody else who can answer this or shed his ideas. I don't want perfect answers. Anybody who thinks anything of it can answer it. Like, let's hear from, we have another hand, Awais, Ahmed, Amr, Badr, you guys need to participate as well. Hamza, Jawad, any answers that you can think of. And Anderson, I'll be sorry if I'll have to say that you are not coming with a mic. So if you don't have an active mic, you can participate through a chat, but you're not even chatting. So let's give the mic to Awais. Let's see. Yes, Awais, you are absolutely right.
Mister, one of the factor is temperature.
Perfect. And what's the second?
pH.
Perfect. So good job. These are the 2 factors on which the enzyme will affect. Enzymes, concentration, we didn't discuss this, so let's leave it. Temperature and pH. So lock and key model. What are those lock and key model? Yes, Yusuf, you're raising a hand.
Mister, that was a long time ago, but I have a question.
Yes.
I want to answer. One of the functions, it's a catalyst. the
One of the function of catalyst?
that one of the function of enzymes, they're catalysts.
They are living catalyst, yes. They are speeding up the chemical reaction. Good.
Yeah,
And
that's
what about lock and key? Anything about this?
Wait,
very
I'll
simple.
think.
It's very
It's
simple.
about like when, right?
Yes.
I'll answer later.
Try it, no problem. And he put himself into. What is pH? pH always is something which is acidic in nature or alkaline or basic. Like milk is basic in nature and lemonade is acidic in nature. But you want to know that how strong are they? How strong is lemonade, and can we have a stronger acid than that? Then we have a pH scale which measures that how many hydrogens there are. So the more hydrogen it has, the more stronger acid it will be. The less hydrogen it has, the stronger basic it will be. Okay. We might study about the pH in more detail in chemistry once you will realize that what makes a liquid acid and what makes it a base. So once we understand that, then we'll be able to know the theory, the working phenomena of the pH. So let's go back to the lock and key model. It's very simple. Mansoor is raising a hand, so let's see Mansoor. Sorry, I muted you again.
Mister, I forgot to lower my hand.
Okay, no problem. So I'll lower everybody's hand and we'll wait for the Ghazi to come back as well. Lock and key model. Okay, Basim is raising. Okay, yes, Basim.
Mr. What is lock, key, and model?
No, it is lock and key model.
No.
Abdullah Rafa, this is quizlet.com. It is not Nearpod. And Basim, the lock and key. Like every lock has one key, and once they are coming together, they fit in like a jigsaw puzzle, right? So the enzyme works like a jigsaw puzzle. If the edges meet together, then they would work together. The model of the enzyme that shows the substrate fitting perfectly into the active site. If you remember, we saw that in the presentations and in the videos that the active site has a shape. And if the substrate comes with the exact shape, just like a jigsaw puzzle, and if they fit together, then they will work. Otherwise, they will not. Induced fit model, we did this, but I'll skip it for the moment. And no. All right, so that's it for the revision session. We'll go to the video. Ahmed Al-Hazmi has a question. Yes, Ahmed? You have the mic, Ahmed?
How many homeworks do we have on science?
Every week you have a homework. Week 2, week 3, week 4, week 5.
Mister,
Yes. Hello, can you hear me, Ahmed?
no, and this week, how many homeworks do we have?
One homework every week. Right?
Oh, Mr. and module, what?
Module is called week 6. We are on week 6. So we'll have homework for week 6. And we have Basim raising a hand again. Yes, Basim? Basim, you have the mic?
It was from a long time ago.
Okay, sorry about that. So let's go to the video. Let's see the small video. Counselor is here. We're going to watch the video. It's about 5 minutes. And if you have any questions related to it, please raise your hand. I'll be with you guys, and I'll try to explain what is happening over here as well.
Enzymes. Human body is a product of different chemical reactions and processes. But what controls these reactions? In 1833, a French chemist, Anselme Pien, was the first to discover the vital force that drove these reactions and named it enzyme. Enzymes are substances, proteins, or in some cases, ribonucleic acid, RNA, that speed up a biochemical reaction by modifying specific substances called substrates. Enzymes are supremely selective in who they bind to and modify, hence their specificity. Mode of action. Easier said, but how does a tiny enzyme speed up a chemical reaction? The answer to this is simple. Enzymes perform this critical task by lowering a reaction's activation energy, that is, the amount of energy needed for the reaction to begin. Enzymes bind to their substrates, hold them in such that chemical bond breaking and bond forming processes take place more easily. Enzymes have a specific place in them called active site, where the substrate binds and real-time action takes place. Active site has a specific size, shape, and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme's active site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Cofactor.
All right, this is the example of the lock-in key over here. The way they are adjusting to each other, and nothing else can come in between them. If the shape matches, then these substrates will be able to go in. This is called the lock-in key model.
Thanks to these amino acids, an enzyme's active site.
You can see that this substrate is trying to enter into that enzyme, but the shapes don't match, so they will not be binding together. This enzyme will not be able to work for this one, for this substrate.
Site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Cofactors may be cations, positively charged metal ions, activators, bound temporarily to the active site to activate the enzyme, organic molecules, vitamins or vitamin products, coenzymes that join enzyme substrate complex temporarily, prosthetic groups, permanently enzyme bound. Many enzymes only perform their catalytic role when associated a coenzyme. An inactivated enzyme, protein, along with its coenzyme, non-protein, makes up a system called holoenzyme. To add to
this is, let me keep it very simple for you guys. The enzyme over here is inactivated. It is not active right now. To get it in the activated state, we need a coenzyme. The coenzyme will activate, will turn it on. It's just like you need an ignition. So once they're ignited, it is called an activated enzyme. Instead of holoenzyme, you can say it is an activated enzyme. Yes, Mr. Ghazi.
Mister is like when an active enzyme is like the key of to enzymes to work.
Yes, exactly. It's like a key, so you have to ignite it, you have to turn on like an engine, and then it will start working. So
Yes.
without a key, the engine is not activated, and they will not be working. Okay, Abdullah is also raising a hand. Yes, Abdullah.
Yeah, just a question about things like, for example, some foods have certain chemicals that the enzymes, react to. What happens to those enzymes when somebody doesn't eat, a specific food? For example, somebody doesn't have, doesn't take sugar, like doesn't take carbs or any sugar, and the enzymes that would, usually eat sugar. What would it do in that? What would it do in that case? Would it die? Would it just like be there?
Yes, they'll not be dying. The only time they die is when we are sick. Then we have to replace. Oh, that's loud. So we have to replace them. No problem. So we have to replace them and get the new ones. Otherwise, they're going to stay in activated state.
Yeah.
They will be inactive. They will be there. They will not be doing any function because they can't find their own type of substrate. Like you said, like a person is cutting down all the carbohydrates. So the enzymes would be relaxing, would be not doing any function. They'll be sitting by themselves. Ghazi, yes, you have the question?
Mister, like an insulin is like the active site for the cell to enter the glucose. Yes.
Insulin actually is like an activator for the cells to let the sugar go into the cell. It's like a
Yes.
key. It's like a key which opens up the door of the cell so that sugar can go in.
The glucose can enter, yes.
Yes. Otherwise,
It's the
it
same like,
yes.
Mr. Okay, the active site of like an enzyme, it's made what substances? Like it's glucose or what?
1
made
minute.
of?
I'm sorry. I got distracted by another good question. Amar, he asked a very good question that if you are sick and your enzymes are dying, but you get a new one, yes, you do get a new one, but it won't be instant. You might take 2 to 3 days or 4 days to recover. That's why you feel a little bit weak. After you're being sick, you take a little bit time to recover. So yes, you will get the new enzymes, inshallah, but it takes time. Yes, Ghazi, you were question, you were asking.
Mr. Look into that, please.
Look,
Yes,
what?
Mr. I want to tell you something. Allahumma salli ala Muhammad. Mr. Like, what is the active site of an enzyme made of?
The active side is what of enzyme made up of. They contains usually the ion side, the positive and
It's
the
like an
negative.
iron.
They have a lot of atoms surrounding this active side with the positive and the negative ions. And when the substrate comes in, it has the exact opposite of it. So that's why they attract it, they get attached to it. The positive will attract the negative and the negative will attract the positive. So imagine that this layer, let me draw it over here. Imagine you have the layer over here with all the positives, positive, positive, 2 positives here, and then negative, 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 positive. So this combination should appear over here. But the substrate comes with the exact opposite combination, which is over here, and they'll attract each other and they'll make a bond. So let's go and let's complete the video. We'll continue the questions after this.
To the complexity, scientists termed this inactivated enzyme as apoenzyme. And therefore, the equation becomes holoenzyme equals apoenzyme plus coenzyme. Models of enzyme action. Lock and key hypothesis. This is the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form a reaction intermediate. Just like the key fits in its specific lock, the shape isn't changed here. Rather, the structure of the substrate absolutely complements the structure of the enzyme, like puzzle pieces. Induced fit hypothesis. In this model, the enzyme, upon binding of substrate, changes shape. The matching between an enzyme's active site.
Abdullah Rafa, can't you hear the sound of the video? No? Let me check again. The sound is on. How many of you can't hear? Please raise your hand. Awaz can hear. Ghazi can hear. So check, Abdullah, check your voice and speakers or the headphones. Okay, Abdullah is trying to. Yes, Abdullah.
I'll leave and join again. Hopefully it'll work then.
Okay, let me rewind a little bit. Can you hear the sound now? Check.
Enzyme's active site is unique only to a particular substrate. In addition to the active site.
Sorry?
Maybe because I wasn't in screen share. Yeah, I can hear it perfectly now.
All right, good. Okay.
Many enzymes consist of a non-protein part called cofactor. Cofactors may be cations, positively charged metal ions, activators, bound temporarily to the active site to activate the enzyme, organic molecules, vitamins or vitamin products, coenzymes that join enzyme substrate complex temporarily, prosthetic groups, permanently enzyme bound. Many enzymes only perform their catalytic role when associated a coenzyme. An inactivated enzyme, protein, along with its coenzyme, non-protein, makes up a system called holoenzyme. To add to the complexity, scientists termed this inactivated enzyme as apoenzyme. And therefore, the equation becomes holoenzyme equals apoenzyme plus coenzyme. Models of enzyme action. Lock and key hypothesis. This is the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form a reaction intermediate. Just like the key fits in its specific lock, the shape isn't changed here. Rather, the structure of the substrate absolutely complements the structure of the enzyme, like puzzle pieces. Induced fit hypothesis. In this model, the enzyme, upon binding of substrate, changes shape. The matching between an enzyme's active site and the substrate isn't just like two puzzle pieces fitting together. Rather, the enzyme changes shape and binds to its substrate even more tightly. This fine-tuning of the enzyme to fit the substrate is called induced fit. Environmental effect.
So the induced fit is where it is not 100% having the shape needed for the substrate, but it can actually adjust itself a little bit so that it can accommodate. So you can see that this is not 100% shape of the substrate. So it is a little bit flexible. It can actually, we have 3 hands up. Yusuf, yes?
My question is, when an enzyme is made, does it start as the shape as the thing, or does it bind to it
I don't understand your question. Can you repeat it again? Yes.
When an enzyme is made, like if a new enzyme comes,
Yes.
is it made with the shape of the
With the active site? Yes.
Yeah.
Yes.
Of the thing, or does it like every time it sees it, it turns into it until?
No. They have an active site. The DNA, when it sends out the message that it gives it to the ribosomes that we want to create an enzyme. Let's say we are trying to create amylase. That is for the sugar, for the digestion of the starch in our mouth. It will have one kind of active site. And what we are discussing right now, the lock and key, it's a theory. The induced,
I know.
It's a theory as well. So we haven't completely understood that how the active sites actually work, but they are meant for one type of substrate. So one of the theories says that it has a perfect shape and it should match like a jigsaw puzzle. The second theory says, no, it is adjustable a little bit. It has a few, gaps to fill in. So once they receive, they adjust themselves about 2 to 3%.
Doesn't the coenzyme help if there's gaps?
Exactly. This is what the theory says, that the enzyme is being helped by the ions and the coenzymes to get induced fit. It definitely gets help. And they get the recognition of what type of substrate is it coming. Is it a protein? Is it a starch molecule? Or what is it? So it's a complex science, and we are just discussing things in a very simple manner. So Awais has a question. Yes, Awais?
Mister, when he said holo enzymes equal not activated enzymes plus coenzymes, what did he mean by holo enzyme?
By the holo enzyme, it meant that it is now an active enzyme. The active enzymes, how can we get an active enzyme? You have to have an inactive enzyme plus coenzyme. So once we unite the coenzyme with the sleeping enzyme, the inactive, it becomes an active, somebody who's awake now, right?
So it starts
working. Yes. It's just like you're sleeping and somebody wakes you up and gives you a cup of coffee and you're all up and active, just like that, exactly the same. So we have a sleeping Awais and we have an active Awais. What makes you active? Let's say coffee. Right?
Okay.
Okay, perfect. Abdullah Rafa is asking a question. Yes, Abdullah.
That's from a way long time ago, like from the beginning of the lesson. Sorry about that.
Okay, and Ghazi is asking a question. Yes, Ghazi.
Mister, can you repeat the part of what always was talking about? Because Mr. Alhamdulillah, the kid is now go. They are safe, Alhamdulillah.
Who is safe?
The kitten. My kitten tried to suicide, Mister.
Oh, really?
Yes, Mister. We jumped from the third from the stairs. She jumped from the stairs.
Okay, so we are left with only 5 minutes. So Ghazi, I hope you can see that video on the canvas.
Okay.
And if there's a question, let me know.
Substrate even more tightly. This fine-tuning of the enzyme to fit the substrate is called induced fit. Environmental effects on enzyme function. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include temperature. The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affects chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes. pH. Amino acids present in the active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrates to bind. Extreme pH values can denature enzymes. Enzyme concentration. Increasing enzyme concentration will increase the rate of reaction, as more enzymes will be available to bind with the substrates. However, after a certain concentration, any increase will have no effect on the rate of reaction. Substrate concentration. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules, so more product will be formed. But again, this effect is valid up to a certain concentration. Inhibition of enzyme activity. Some evil substances called inhibitors reduce or even stop the activity of enzymes in biochemical reactions. They either block or distort the active site, thus inhibiting the reaction. Based on this, there are two types of inhibitors given below. Competitive inhibitors occupy the active site and prevent a substrate molecule from binding to the enzyme. Non-competitive inhibitors attach to parts of the enzyme other than the active site to distort the shape of an enzyme.
Okay, so we have questions. Let's pause the video. Yes, Ghazi.
Mr. In the Death Valley in the United States, the temperature was before 1 month, 2 months, 55, the highest temperature in the world. How, like, Mr. The people who live in the Death Valley used to be like in 50, 51, 52. How, like, the enzymes of them work?
The enzymes will be affected. They will not be able to work. They will be denatured. They will be exhausted. They will be tired. They will be stressed. That's why the doctors say you'll have to take rest. When the temperature goes really high, we actually stop working and the schools are closed. Even in Saudi Arabia, we had a temperature once about 55 degrees, and the roads, the cars started to, the radiators were jammed up and everybody was stranded. So that human body becomes stressed. So if this stays for a longer period of time, this can leave very drastic effects on the body.
No, Mr. It take only for it's been for only 55 only for 1 minute and 30 seconds.
Yes, but if it stays for an hour or two, it can leave a very bad effect on the body, seriously bad effects.
And Mr. I have another question. The enzymes that incorrupt the enzymes or the active enzymes, what are they made of? Also protein or iron, I mean?
1 minute again. I got a question which got my attention. Tahir, it will slow it down, slows it down. Yes, what's your question again?
Mr. The X enzymes, the X molecules that the red one in the video are made of what?
The inhibitors, that they can be a bacteria or they can be food poisoning substances. It could be metal ions as well. Junk food usually have them as well. So a lot of junk food contains particles which blocks our enzymes. The enzymes especially, which tells us that you are full now. So you can eat more. And they would sell more. Make sense?
Yeah, it makes sense. Thank you. Thank you.
All right, thank you very much, Hadi. So unfortunately, the time is up, and Alhamdulillah, we had the proper recording this time. I'll stop the recording.